So let's take the steps of translation one at a time. So here we can see the brown. This is our messenger RNA. Our ribosome has come together on our messenger RNA. And the first codon of our messenger RNA is our start codon, and it's AUG. The tRNA that has a anticodon that is complementary to that AUG codon will bind. So the tRNA that's going to bind, and of course these are hydrogen bonds, which remember are individually very weak. You're going to have your tRNA that's complementary to the AUG codon bond. So complementary to A is U, complementary to U is A, complementary to G is C. So this tRNA molecule brings its correct amino acid to this part of the message. The correct amino acid that it codes for based upon our codon table is MET. There is another site on the ribosome which is capable of reading the next codon, which in this example is GUG. The tRNA that has the complementary anticodon to GUG will bind in this ribosome binding slot. So here we can see the next tRNA that has the complement to the GUG, has the anticodon CAC, and is carrying its correct corresponding amino acid VAL. So this second tRNA with its amino acid is going to bind complementary into this site on the ribosome. Now that they're bound right next to each other, a peptide bond can form between the two adjacent amino acids. Because remember, our goal is to make a polypeptide chain, which is made out of amino acids held together through peptide bonds. The next step, notice the ribosome has moved down the messenger RNA. So now the AUG is not being read by the ribosome. So the tRNA that was complementary sitting in the slot where the AUG was now leaves. Notice it's leaving without its MET because the MET has been bound to the second amino acid VAL. So this site right here on the ribosome used to be over here, but now the ribosome has moved down. So now the site that was filled with the second tRNA with its valine is now empty. And the ribosome is reading UUA from the DNA. So the complementary tRNA, complementary to U is A. So its anticodon is AAU. That's complementary to the codon UUA. It's going to come into this spot, temporarily bind, delivering its amino acid LEU. So those two tRNAs sit side by side. A peptide bond forms between VAL and LEU, joining them. And I bet you can guess what the next step is. The ribosome is going to move down to the next codon. As it moves down to the next codon, the tRNA that was holding VAL in place to it leaves because now the VAL is held in place by the MET and LEU. Now we have a new codon, GGG in this example, in our ribosome spot. That will call for the tRNA with the correct anticodon. The correct anticodon is complementary to GGG, would be CCC. So that tRNA molecule comes in carrying its correct, according to the codon table, amino acid GLY. So notice they're now side by side, LEU and GLY, and a peptide bond can form. And this process just keeps repeating over and over again with the ribosome moving down one codon at a time. That process is called, where it moves one at a time, a translocation, until we reach the end of our messenger RNA or until we reach a stop codon. The end of the messenger RNA should have a stop codon, and you shouldn't really have a stop codon anywhere else within the messenger RNA. if you do, then you're truncating your polypeptide and you're making it shorter than it should be. And that could be problematic for the cell. But in summary, the ribosome is going to continue to move down the message one codon at a time until it reaches the end of the message, which is recognized with a stop codon. When that stop codon is reached, the amino acid, large and small subunits will come apart. It will release the message, release all the tRNAs, and release the polypeptide chain. Your end result is going to be a polypeptide, which is why we did this whole thing to begin with. So let me just close out this chapter with a quick summary. This is an excellent illustration showing you in general the processes that we discussed. So here we have our nuclear envelope. Inside the nuclear envelope is our nucleus, and inside the nucleus we have our genetic material, which is represented here as this purple. We have transcribed from our genetic material an RNA molecule. So the process of transcription has occurred. Our RNA leaves the nucleus by way of nuclear pores. So here we have our messenger RNA now out in the cytoplasm. It is going to be read by a ribosome. A ribosome is made up of a small and a large subunit. Here we can see a ribosome has been assembled around this messenger RNA molecule. As the ribosome reads the message, tRNA molecules that are floating free out in the cytoplasm containing the correct amino acid will bind to the complementary codon that is being read by the ribosome, delivering their corresponding amino acid. So as the ribosome continues down the messenger RNA, the process of translation occurs, and our end result is going to be the formation of a chain of amino acids linked together by peptide bonds, and this structure is called our polypeptide, which becomes our protein. Down here we can see an actual image from a cell of a messenger RNA molecule, that's this thin line, being read by multiple ribosomes. That's these dark dots. And multiple ribosomes reading a message at one time are called polysomes. And then these little strings coming off of the ribosome are the polypeptides that are being formed. And why this is important is because we learn the process a step at a time, and it makes it seem like when we learn it that one ribosome reads one message one time, and then for the message to be read another time, we need a whole new ribosome after that ribosome is finished, and that's not the case. A message is being read by a ribosome, more than one ribosome at a time, so that we could get a large amount of our polypeptide formed. So I hope that this presentation has been helpful, reviewing the structure of RNA versus DNA, reviewing the structure of nucleic acids and nucleotides, discussing how to use a codon table in the process of translation, and of course in summarizing the steps of transcription, which is taking DNA and forming RNA, and in summarizing the steps of translation, which is taking our information in our RNAs and making a polypeptide chain.